Okay, welcome back everyone to the afternoon sessions. I think 2:01, so I think we can start this session on subcellular features and then cellular state. So my name is Meng Wang. I'm a senior group leader at 4D-CP at Janelia. So I will be introducing all the speakers in this session. So our first speaker is Jonathan Weissman from Whitehead, MIT, and also a huge investigator. He's going to talk about a multimodal platform using in vivo pooled genetic screen to understand liver functions.
Thank you. So my lab started looking into systematic genetics with yeast, and it was really because that was a system you could do clean manipulations in singles and combinations. And all that changed in the summer of 2012 when Jennifer Doudna came over to my office and said, we have this great programmable nuclease. What are all the things we can do with it? And so together, a few of us there, Jennifer, my lab, Stanley Cheese, took her nuclease the world's nuclease, and broke it so that it didn't cut anything. And what that did was allowed you to put in effector domains. There have been many now effector domains, but the first were either an inhibitory or an activator domain, and essentially gave you a volume switch in which you control the expression of any gene or any combination of genes. And of course, the programmable nature of this, plus the fact that pooled oligosynthesis and massively parallel DNA sequencing had recently come out, allowed you to in a pooled manner where each cell would get a different perturbation, explore in a genome-wide way the relationship between gene activity, whether you turned up or down the gene, and the phenotype. For example, would you be resistant to a chemotherapeutic agent like vemurafenib, or would you turn on unfolded protein response, giving you evidence that you have a protein folding problem in the ER? The challenge with these approaches is that, as Aviv alluded to, you have to decide ahead of time what is the biology you're interested in. So if you're interested, as we were, in folding in the ER, you build a reporter for the unfolded protein response and do your screen. The other challenge is that you have a one-dimensional readout, and of course cells are in some high-dimensional manifold. So you could have two cells in a very different state that had the same level of induction of unfolded protein response. Say, one maybe had a degradation problem and the other one had a chaperone problem. And deconvoluting this is an extremely nonlinear, poorly understood projection. And deconvoluting this is like each gene could be a graduate student career. So that didn't scale. So that motivated us, in a wonderful collaboration with Aviv, to develop the perturb-seq approach, in which each cell would get an individual perturbation, but now you would do single-cell RNA-seq. And with this single-cell RNA-seq, you would read out the perturbation and the consequence of the perturbation. In essence, every cell then became a test tube, and you could read out the consequence of the experiment in that cell, as well as the nature of the experiment in a massively parallel way. And then mapped the effect of each of these perturbations in this high-dimensional space, at least to the extent that you can measure it by RNA. We then, Joseph Rapoga, really, and a wonderful student then in the lab, really sat down to scale this from 100 genes to a genome-wide scale. And with this, working together with Ruben Saunders, developed the first genome-wide genotype-phenotype map, in which you would have here each of these columns would be the genetic perturbation. So this is turning down, one gene versus another gene, and the effect on the transcriptional response. You can now cluster genes based on their co-expression networks, and in each of these modules then would be a co-expression, basically a set of genes that are being regulated in response to a particular challenge. So you'd have the unfolded protein response, the heat shock response. Many of these were well characterized, but some of these were just new transcriptional states. And in this way allowed you in an unbiased and principled way to understand the underlying structure of gene expression networks. You could also cluster genes based on the genetic perturbation. So with the idea that two genes that were doing similar function in the cell would cause a similar transcriptional response. In this way, organize the genes based on function, again, with no a priori, even necessarily any knowledge about what that function is. So allowing these sort of principled discoveries. And then finally, and I think one of the most potentially transformational ways is that you can create a synthetic phenotype, an in silico phenotype, based on this genotype-phenotype map, and then do this screen in silico after the fact. And I'll give you one concrete example of that, our work on chromosome instability and aneuploidy. So in this way, we were able to explore a variety of very different aspects of biology. So 3 examples here. One is distinguishing different stress responses, the integrated stress versus the unfolded protein response. Here's an example where we functionally dissected the integrator complex, allowed us to discover a new functional module of the integrator complex and a missing component. And this missing component then allowed Patrick Kramer, and quite generously acknowledging our role in enabling this, to solve the structure and determine the function of this. And then in the final example, we were able to do a genome-wide screen for aneuploidy, one screen that would have been very hard to do by the sort of standard growth or fluorescence-based reporter assays. So you might wonder, how would we do an aneuploidy screen based on transcriptional response? And in fact, there is no specific response to transcriptional response to aneuploidy. So there's no immediate signature that you could say, these 5 genes went up, these 10 genes went down, that looks like aneuploidy. However, if you ordered the genes based on where they were in the chromosome, if you lose, in this case, chromosome 2, what you'll see is a global across chromosome 2, but not the other chromosomes, decrease in expression because you have only 1 copy of chromosome 2, not 2 copies. So in this way, we can make the synthetic phenotype and explore for each perturbation how it's affecting the development of aneuploidy. And here's an example of TTK, a kinase on the spindle pole that is important for checking the integrity of the spindle pole. When you knock it out, as was well known, you get aneuploidy. And you see each of these is a cell. You see these cells lost chromosome 1, these lost chromosome 2, these gained chromosome 3. So there really is no coherent single transcriptional response. But by delving into the data, we can create this entirely synthetic phenotype and do an aneuploidy screen, even though nothing in the screen, we didn't even know when we started that we would be doing an aneuploidy screen. And I say this, why did we do this? In large part because we were really strongly influenced by my friend and colleague, Angelica Mann, who had done beautiful work on aneuploidy. In fact, Joseph's brother was a graduate student in Angelica's lab. So that's why we got into it. But I think this is an incredible opportunity for the sort of agent-based AI, where you could, and I did the test, I asked, how would we test for aneuploidy from single-cell RNA-seq, and it gave me a very nice answer, and with all the references of how you would do this. So you can imagine an agent that would you'd set on the data and say, let's explore all the different aspects of cell biology and find out which ones we could get from single-cell RNA-seq, and go in, pull out the data, and pull out other data sets and do the screens and hand you the answers. The other thing that this has been very useful is in virtual cell efforts. So this has been sort of almost a unique data set that will change quickly, or is changing quickly, but for efforts to develop virtual cell or predictions and silico predictions of the impact of perturbations. So all this is terrific, but these were all done in cancer cell lines. And I'm haunted by something I heard at a functional genomics conference, which is you don't want functional genomics to get you to the wrong answer faster. And so the models and the measurements that you make are critical. And cancer cells are great for some aspects of cell biology, but they simply can't recapitulate that metazoan cells live in communities. And my sort of background from the biology is on protein folding and homeostasis and stress response. And I was acutely aware of the fact that you're working with these cancer cells under completely non-physiologic, not the normal conditions they were meant to be grown in. So we really, I think we as a field feel you have to move away from monocultures of cancer cells in a dish. And there's nice systems like organoids and other assemblies or IPS-derived cells. And those are powerful systems and certainly have their role. but we really wanted to be able to discover regulators of complex physiology. So things like development, metabolism, especially in the context of liver, inflammation, disease, and cell non-autonomous effects as well. This motivated. So in order to do this, we really had to do the screens in a true in vivo setting where we could deliver the genetic perturbations, in this case a guide, in a mosaic animal in which each cell would be getting a different perturbation, be able to measure the phenotypes that capture the complexity of the in vivo physiology. We'd have to in situ be able to determine the genotype, determine in each cell whether it got a perturbation, and if it did, what the nature of that perturbation is. And then finally, and this is really the most open-ended aspect of this, is develop analysis packages and strategies that can pull out and interpret these complex phenotypes. So I said this was really a wonderful effort by Will Allen, who's in the audience, who was then a junior fellow working with Jiawei Zhuang, and Ruben Saunders, who was then a graduate student, has now become a junior fellow and is working with Jiawei and me, and we're all continuing to work together. So it's just a terrific team that brought the expertise needed to bring this together. So this then, basically the approach we developed, we called Perturb Multi, because it gives you a multimodal readout. We would deliver genetic perturbations, in this case the mouse, but you can do this to other tissues, including the heart and the brain and lungs. You create a genetic mosaic in which each cell would get a different guide RNA. You'd wait for the animal to become an adult with an AAV by a Cree system, turn on the Cas9, so turn on the perturbation acutely, wait, for example, 10 days for the perturbation to take effect, and then perfuse the animal to freeze the cells in situ in that state. And from this, we can either disassociate the cells and do a single-cell perturb-seq experiment and with a full transcriptome readout, or take sections and image, in which case you can get high-resolution images of multiple different proteins, as well as measures of the RNA and the guides. And in this way, we would get these multimodal readouts of the subcellular morphology, gene expression, cell organization, cell interactions into an integrated data set. And the other thing that really came as a surprise to me, which is when you thought about it, is that there are a lot of cells in a mouse. So in a mouse liver, you get 100 million cells or so, hepatocytes. And compared to the cost of the media and the effort that you have to go in to do, for example, iPS screens or even cancer cell screens, let alone organoids, the mouse, you just give it mouse chow at $1 a cage, cage charges, and it ensures that it keeps it at the physiologic conditions. So it's one of these happy circumstances where not only are you able to do the experiment you want, but can actually do it in many ways at a bigger scale and more cost effectively. So just to get a little more granular, the basic approach was you would take fixed tissue, and really the key that Will and Ruben worked out was that they could both disassociate the cells so that they could do the single-cell RNA-seq at very large scale. And I should say there's been really a transformation in the scale in just the last year or so that we can do single-cell RNA-seq, but then also that you can do the imaging through hybridization-based approaches. And this is, looks like a simple slide. Over a glass of wine, me or even better Will can tell you what he did to get this to work. And if you give us a bottle of scotch, we can start talking about all the things that didn't work along the way. But it was really the key was to get these all to play together so you get these true multimodal analysis. So basically, what one would do from the imaging part is take slices and then go through these rounds of combinatorial readout that would let you do gene expression, morphology, or readout of the guide barcode in the same cell. And as I said, our sort of test bed here has been the liver. The liver's kind of extraordinary organ. I've learned a lot more about it along the way. It's, of course, responsible for much of your metabolism. It's the central dispatcher and mediator of energy and many of the key metabolic processes. It also has this organization, of this lobular organization, which there's an outside, the periportal and the pericentral. There's high oxygen on the outside and low oxygen on the inside. And this allows it to separate its different functions. So like oxygen requiring processes like beta oxidation or gluconeogenesis and protein secretion occur on the outside, whereas that glycolysis and lipogenesis occur on the inside. And this is this zonation is next to impossible to really recapitulate in growing in vitro. So I'm sure you've heard about the people with primary hepatocytes, and they can keep them going for a while. But this high secretion, this organization from high to low oxygen are just things that you can't physiology just nearly impossible to access in vitro systems. So when one does these experiments, you can take the single-cell RNA-seq and imaging data and get these sort of abstract UMaps of different cell types, and we can see that we can clearly identify and recapitulate the full spectrum of cell types. But you can then, of course, because you have the imaging, you can then place them in their true three-dimensional structure. And you can see here for the hepatocytes, this pericentral and periportal structure, and then the non-hepatocytes overlaid them. But also, we can zoom in any individual cell and look, because we had 14 different protein and 4 RNA markers, we can look at high resolution what the ER looks like, what mitochondria looks like, what the state of TOR signaling is, look at pre-mRNA versus mRNA. And basically, any thing for which you can have a validated antibody. And I tell you, the key limitation is having the validated antibodies, which hopefully as a community will all increase. But because the antibodies are encoded in an oligo, there's no limit to the spectral separations. You can just decode them by the Murfish combinatorial strategy and have, in principle, an unlimited principle an antibody for every protein if they existed. So, of course, the image analyzing images is not nearly as straightforward as RNA. So, when do two images look similar to each other? And this has been an effort from Jingzhe Pan in Xiaowei's lab using an autoencoder embedding approach that allows us to compare the similarities between the images. But I say this is really a work in progress, and one of the places where AI is just going to be not a layer on top, but absolutely essential for the analysis of these type of data. But with this, we can, as we did with the genotype-phenotype maps in vitro, we can, for each perturbation, measure its transcriptional response and its response to these 14 proteins and 4 RNAs, and then order perturbations by their similarity, either by imaging or by the sequencing phenotype or the imaging phenotype. And you can see that there's clear similarities between the two, but there's also strong differences. There's some things that are picked up much better by the imaging, and some things that are picked up much better by the sequencing. Again, sort of hinting at the value of having this these multimodal measurements. So as with our in vitro cancer cell genome-wide perturb-seq, we're able to get in this smaller scale in vivo perturb-multi, we're able to measure lots of different aspects of biology, like the UPR versus ISR, the things that are regulating secretion. So the liver is pouring out albumin. It's 50% of the proteins in your serum. And we see that what's controlling the production of albumin, the biggest hit, of course, is albumin itself. You knock out albumin, you don't get the protein. You don't get expression of the gene or the protein. But the second biggest is cell 1, which induces the unfolded protein response. And this was really gratifying result because years ago, Julie Helene in my lab had discovered that one of the aspects of the unfolded protein response was not just to turn up chaperones, but to turn down secreted proteins, relieving the burden on the ER. But in our in vitro systems, we're getting 20% effects. And we put together a story that this should be important for relieving the burden of it, but it was hard to understand. Was this really an important aspect of how cells under physiologic conditions responded? And we see here there's really a massive downregulation of albumin in response to ER stresses. And similarly, we can do a screen for things that are perturbing TOR activity. We can also look under different physiologic conditions. So the liver undergoes massive physiologic changes as you fast or refeed or give high-fat diet. We can see in the fasting versus ad libitum, most of the genes have a similar effect, but a set of genes here really dominated either by the TOR or by the autophagy genes become much more important and much more interconnected under these fasting conditions. Again, now giving us a chance to understand how TOR and autophagy are working under true physiologic condition. And then finally, as my last vignette, I'll talk about hepatic steatosis. So the liver can get filled with lipid droplets, and the steatosis can go on to cirrhosis and cause so-called MASH or NASH, which is a major health concern and great interest in pharmaceutical because of the increased levels of MASH and its consequences, either in liver failure or liver cancer. And that the reason this is that the liver is taking up fatty acids from the blood and then using it and then also producing de novo fatty acids, and then it either puts some in, goes under lipolysis to use them for energy, stores triglycerides as VLDLs to export, or creates beta oxidation, undergoes beta oxidation to give you ketone bodies, which then go to the blood. So both VLDLs and the ketone bodies go to the blood, and they basically, for example, under starvation conditions, feed your muscle, feed your brain with sugar, with glucose, et cetera. So this is its role in the dispatcher as a dispatcher. But if there's an imbalance between this, if more fat is taken up than is given out, you end up with this fatty liver disease and the steatosis. So we, as part of this, because we had perilipin, which is a marker for the lipid droplets, we had done a steatosis screen. And you see things like mTOR really decreased steatosis, whereas these genes, EIF, INSEG, AARS, and PTEN dramatically increase steatosis. And you look at these genes from the imaging phenotype, and they all look pretty much the same. So these are random cells that have knockdown in INSEG or AARS or PTEN, and you see they're basically filled with lipid droplets. On the other hand, because we had the genome-wide perturb-seq, the single-cell RNA-seq, we could ask, how did these phenotype of these genes compare when we looked at it from the transcriptional response? And we see that they break down into three very different, really uncorrelated transcriptional responses. So if we had only had the imaging, we would say these are all steatosis genes. If we had only had the transcriptional response, we would say these are completely unrelated genes. And it's only through these together that we're able to see that there's multiple different pathways and different processes that combine to lead to the steatotic phenotype. So where are we going? We want to increase scale, increase tissues, go to different animals, et cetera. But also really, this is, I hope as I've illustrated, multiple different levels. The critical thing is going to be to use machine learning to interpret the data, use it to these agents to explore the data, and then use the data to train models that will then drive discovery. So with that, I'll thank the people. Thank you for listening and the people who did this. I've mentioned these guys along the way. They're really the heroes behind this, with Jaspeed and Jing Jie, and then Britt Adamson and Tom Norman pioneered the original perturb-seq work from our side, and Joseph Rapogo really scaled this up and led with Ruben the genome-wide perturb-seq. Thank you, and take any questions.
Thank you, Jonathan.
Thank you. Very nice talk. So in your Pritov-6, when you have the ER stress increased, and I've seen that you have like 6 hepatocyte subpopulations, I think. So do they get different effects? For example, for albumin secretion?
So there's definitely, this is a good point, is that both from the imaging where you actually know where you are physically and from the single-cell RNA-seq where you can determine which state you're in, you can look at the effect in each of these different zones, or the 6 different zones. And then, since, the beta, as was known, albumin secretion is really more on the periportal side. So the pericentral was low as it should be, but the periportal really went down. But we could see that it wasn't just you were losing periportal cells, that the periportal cells were failing to produce albumin. We also in this actually did a screen effectively for zonation, which is another story. Yeah.
Jonathan, it's mind-blowing to think about what you can do with these data. My question was, is the mosaic that you're generating sparse enough that you can then look for a guide and then ask what is the effect on gene expression in neighboring cells?
Yeah, absolutely. So you can control the multiplicity of infection. Plus, because you have so many cells, each guide is going to be in multiple different places. So you get many examples of each guide. So even if there was some confounding that sometimes you're next to another perturbation, you can essentially average this out, essentially do the type of compressed sensing strategy that Aviv talked about. But I did not talk about cell non-autonomous effects, but we think that's going to be one of the really critical aspects of this. And we're really, for example, just thinking about making libraries of secreted proteins, because you can deliver guides, but you can also deliver ORFs. And then from those libraries of secreted proteins, try to search for these types of cell non-autonomous effects, which are very hard to screen through in a traditional paradigm.
Very exciting.
Yeah, I have a question for this strategy. Like you talk about hepatocytes, and how about other cell type in the liver, like Kupffer cells?
The delivery, it's a little harder to get the lentiviral delivery to the Kupffer cells, so that you have to have an efficient delivery for that. I think there's, by pseudotyping, there's definitely strategies that you could try to get to different cell types. But you can look also for the cell non-autonomous effects on the Kupffer cells, which of course is one of the things for example, for inflammation that we would be interested in.
Yeah.
Amazing talk and work. I don't have wine or scotch, but I still wanted to ask, what else would be, if you had more time to do the analysis, and like you said, if AI or some tools were around to help you process this data, what questions would you want to ask?
Yeah. So first of all, I think we do need a little bit larger scale data, but we're really actively doing this. I think there's multiple layers, but one and this is something other people have touched on. What is the true dimensionality of the cell? How much can you infer one from the other? So I gave you examples where the aneuploidy was not really a transcriptional response, and it wouldn't be. So you had to look into, but it was hidden in the transcriptional data. So that's where an example where it's really an algorithmic thing. And then the steatosis, where it wasn't obvious how you would go from transcription to steatosis. But this can now be done really in a much more systematic way. So what is the mutual information between the different types of channels? And that should also then allow us to go back and design the experiment. So we don't want to have to measure everything. And I think this is just, philosophically, much of this is going to be, what is the data set we need to train the model? And the model that we want to train should be the leading thing, and then we design the scale of the experiment around that.
This may be an extension, but Piwa Lee said, can your framework characterize metabolic and immune zonations?
So, I think for sure we should be able, for sure we're seeing the metabolic zonations, and that's intrinsic in this. And, one of the things that we saw, for example, was genes that were impacting the zonation, and in large part they were genes that were known, some that weren't known. But what we found was that they were really acting cell autonomously. So a gene that would get rid of the periportal, and bulk get rid of the periportal zonation, you would see that they would still be physically in the place where there was periportal. So that cell ignored where it was and became pericentral. And I think that's actually important when thinking therapeutically, because this now really shows you just how dynamic the organization of the liver can be, and how you can think about things that would, for example, if you wanted to go into lipolysis instead of fat storage, you could push towards pericentral.
Thank you.